Hello everyone, myself Mahreen.
And myself, Astha.
And today we are going to look at the overview of gene database, which is developed and maintained by the National Center for Biotechnology Information, that is NCBI. This is the homepage of NCBI. Now let's access the gene from the drop-down menu. Or you can also directly access gene from the popular resources. Now, this is the home page of gene database. Now, gene database integrates gene-specific information from multiple data sources. NCBI reference sequence, genomes for viruses, prokaryotes, and eukaryotes are the primary foundation for gene records in that they form the critical association between sequence and a tracked gene upon which additional functional and descriptive content is anchored. Now let's look at one of the example of gene, that is JAK2. JAK2 is also called as like Janus kinase 2 gene. This gene encodes a non-receptor tyrosine kinase that plays a central role in cytokine and growth factor signaling. So here, at this moment, there are like 1,746 individual gene records referencing JAK2, of which 517 belongs to Homo sapiens, that is humans. Now, let's click on the first gene that belongs to human. Now, because this record is so large, note the table of contents given on the right hand side and additional links to related National Library of Medicine databases and other genetic resources. Now, the first section, that is summary sections, opens with the official symbol of a gene here, that is JAK2, official full name of the gene as defined in the public database of the Human Genomes Organization, that is Gene Nomenclature Committee. Now, however, as you can see from also known as a list, that this gene is also referred to as JTK10 in other publications. Now, this gene encodes for a protein, and the summary includes information on how that protein functions and potential issues with gene mutations. Now, this next section of genomic context describes where a gene is located. So in this case, the gene is located on chromosome 9, the Q or long arm, Q or short arm in region 24.1. Now, gene includes information on both the current and past assembly to assist researchers. Now, note that the JAK2 gene starts after the 4.779 millionth base pair on chromosome 9 in this current assembly. This diagram shows the direction in which the gene is read. Now, remember, the double helix can be read in either direction, and convention states the chromosome be presented with the shorter arm on the top or to the left. So JAK2 is read towards the right, but this nearby gene insulin like 6 gene is read towards the left. Now, this next section provides a type of gene browser that offers a tremendous amount of information. So click on this green line, which is representing your gene basically. Now here you can see information on the gene in the green, information on the mRNA which is indicated by blue line, and the red line indicates the translated protein of our gene. Now, the next section showcases the expression of that gene in different tissues of individual. So in our case, JAK2 shows ubiquitous expression in appendix and as well as heart with RPKM value of 11.3 and 11.1 respectively. It also shows expressions in other 24 tissues. The next section, bibliography. needs no introduction. Now, these are selected PubMed citations focusing on a specific function of focusing on the human JAK2 gene. Now, if you need citations focusing on specific function of a gene, then this section on gene RIF, that is gene references into function, can be very helpful. Now, these all links will take you to the PubMed. Now, the next features of gene database will be explained by my dear friend Astha.
Now, coming to the next part, which is the phenotypes. The links here, there are links given here, and the first link will lead you to the BioGrid. This is a database, and the BioGrid stands for Biological General Repository for Interaction Datasets. It is an open access database resource that houses manually curated protein and genetic interactions from multiple species, including yeast, worm, fly, mouse, and human. CRISPR screening facilitates discovery of key genes or genetic sequences that elicit a specific function or phenotype for a cell type. So looking at the screen over here, there are information given about our gene, like author, source name, cell type, cell line, phenotype, condition if any, library, enzyme, and the hit score. Going back and also going to the next section, which is EBI GWAS catalog. It is a curated collection of all human genome-wide association studies that will provide links to various publications related to the gene of interest. Now, going to the next section, which is variations. And as the name suggests, it will lead to a database with variants of our gene. Let's click on the first one, which is the CleanVar database. So this is the CleanVar database page. It is a public archive with free access to reports on the relationships between human variation and phenotypes, with supporting evidence. The database includes germline and somatic variations of any size, type, or genomic locations. So the information over here about our gene is the variation location, genes involved, protein change if any, conditions, clinical significance, and review status on it. Going back. And in the variation section itself, there is another link. We will open it, and this will lead to DBVAR database. So DBVAR is a database of large-scale genomic variants. DBVAR includes events such as insertion, deletion, and inversion of our gene. So coming back, and also going to the next section, which is the HIV-1 interactions. So this has links to publication on HIV interaction with our gene. Here there is the title of that one publication. Its title is Monocyte-Derived Dendritic Cell from HIV Infected Clinical Samples Downregulated JAK2 Expression. This will open in PubMed. Now going to the next section, which is pathways from PubChem. This section provides pathways related to our gene. There are 91 pathways that involve our gene. Let's click on the first, and this will lead to the PubChem, which integrates pathway related information as well. So a pathway involves the interactions between chemicals, proteins, and genes. Some pathways are known to directly involve in diseases. This integration helps to highlight available pathway resources, as well as to provide a context for a given chemical, gene, or target. So here, hemostasis is the pathway, and the pathway is given source, taxonomic scope, taxonomy, category dates. And here are the contents of this page. You can access to any of these directly. For example, the diagram here.
So that's it. This was a brief overview of gene and its associated databases.